process of aerobic respiration. Chapter 5, right? So chapter 5 deals with metabolism. And we were talking about the breakdown, the catabolism of glucose, using glucose as our model example of how materials are broken down to get energy. So glucose broken down eventually to give energy to the cell, right? So release of energy. And when we break down glucose, the process that we're discussing right now is aerobic respiration. When an organism is breaking down materials aerobically, they are using oxygen. And oxygen is needed for the growth of the organism. Right, so that is the reason for oxygen being present. For your exam, we want to review several key enzymes, and these are the ones that we've discussed so far. You need to know their placement in the steps of your aerobic respiration pathway. So if I were to give you a model from your notes, you should know whether or not a kinase, a lyase, synthase, dehydrogenase, or isomerase was involved in breaking down or was involved in catalyzing that substrate, right? So some of the things you want to look out for with kinase, one thing you want to see the transfer of a phosphate, right? So you want to see a phosphate go from one place to the next. And with lyase, the enzyme that lyses will be splitting. So we need to see something split or break. Synthase, to remind you, means join. We need to see something coming together. And usually for dehydrogenase, we want to see our helpers, our coenzymes, NAD plus and FAD plus, which are electron acceptors. And then isomerase, usually what we'll see is going to be a change in the name of the substrate. And isomerases make your isomers. Right, so this term you would need to go over. Isomer would mean that they have the same chemical formula, same written formula, but they look differently. Right? So visually, the structures are different. Right? So these are some of the enzymes that are most important, and if I give you a step of glycolysis or Krebs cycle, you should know how to predict which of the enzymes are involved. It's not so important that you know how you remember every name of in every step. All right? We're not having a test to memorize everything, but we want to understand how does the enzyme change a material. So that is our goal when we're studying our exam. In last class, we covered glycolysis, and I just want to go over some of the models that you will see in your textbook as well. So some of the slides here you may not have in your notes, but you'll see some of the ones that you do have. So the cell keeps a net total of 2 ATP at the end of glycolysis. In the beginning of glycolysis, remember glycolysis is how many steps? Yes, a total of 10, all right? So the activation or preparation will be 10 steps, all right? So if 10 steps, how many enzymes? It will be if 5 steps, how many enzymes? So it will be 5 enzymes. Okay? And then the conservation stage, which happens later, is also 5 steps. So this is our total of 10 steps. All right, so altogether 10 enzymes, but 5 in the activation and 5 in your conservation stage. Right, so in this process, if we look at it in the activation, we use 2 ATP. So the cell uses this. It's going to waste it, but in conservation, it's going to make. All right, so the cell gets energy. All right, so it will make 4 ATP. So altogether, if we subtract the 4 that it gained minus the 2, we will get a net gain of 2 ATP. All right, so this is something that we talked about. I do want to go over some of the representations here as far as glucose. So for glucose in our model, we learned the structure of glucose, and for our model we represent it a certain way. And we use our dots, our circles. So how do we represent glucose? Okay, glucose should have 6 dots. All right. So when glucose is made, we use 6 dots to represent what? 6 carbon. And those carbon will be split in this process of glycolysis. Glycolysis to split a sugar, lysis to split a sugar. So we'll split it into 2 molecules. All right, so we have 1 pyruvic acid. And here is the other one. So we've taken the 6 carbon molecules in glycolysis, again, glyco meaning sugar, and lysis meaning to split. We split it into 2 molecules, 2 pyruvic acid. How many carbon does each pyruvic acid have? Each one will have 3 carbon, right? So we use our dot or circle representation to indicate that, all right? So you should know for the exam what happens, or overall, what is the summary? What happens at the end of the first 5 steps? Well, we make 2. So after the first, glucose is converted to 2-GP, right? Glyceraldehyde 3-phosphate. All right, so this is the summary. And when this happens, we're going to use, right, the 2 ATP, right? So that way we can split, right? We need some energy to split. But in the last 5 steps, the 2-GP are going to be converted into 2 pyruvic acid. And when this happens, NADH will be made, as well as your 4 ATP. Right, so that's the summary of glycolysis. So when they are talking, if you're looking at this model, when they're talking about 2 ATP, right, they're talking about the net. Right, so the net, what is the net total? Right? And when we look at this, oh sorry, I should have put 2 NADH. When we look at this amount that's made, remember there's 2 glyceraldehyde phosphate. That means that 1 glyceraldehyde phosphate makes 2 ATP. Let's make this clear. 1 glyceraldehyde phosphate will make 2 ATP. All right, so if we have 2, then we get 4 ATP. And 1 of the glyceraldehyde phosphate will also make an NADH. Right, so if we have 2 glyceraldehyde phosphate, that means we have 2 of the NADH. And you can see it here, the 2 ATP refers to the net, and the 2 NADH means what is made overall by both of the glyceraldehyde phosphate. After glycolysis, the pyruvic acid will get transferred into the intermediate step. And this step I called a transition step. In this transition step, the molecule of pyruvic acid, right, so pyruvic acid, we said has 3 carbon. So I'll put 3C for 3 carbon, gets converted into a 2 carbon molecule with CoA, all right? So CoA means coenzyme A. So this is coenzyme A. And once the coenzyme A is attached, then the next stage is to go to the Krebs cycle. Right, so now it is ready, the molecule. So this transition step helps to get our molecule of carbon ready for the Krebs cycle. In this transition step, our 3-pyruvic acid will make NADH. And it will also release carbon, right? So if we have 3 here, one of these carbon eventually get removed, and that becomes carbon dioxide. So in the end, the acetyl-CoA has how many carbon? It only has 2 carbon. So that is our model as far as, and it says it here, this is the summary step. So if we look at our model here, so this is what I wrote in. In our model here in the book, well, it tells us 2 carbon dioxide. But here, we're only looking at 1 pyruvic acid. But there's 2 pyruvic acid. If we're coming from glycolysis, remember there are going to be 2 of these pyruvic acid molecules. All right? So I put 3 carbon, right? 3 carbon from splitting your 6 carbon glucose. So that was glycolysis, but once we enter here, each of them have to be converted. All right? So 1 pyruvic acid will give us 1 NADH and 1 carbon dioxide. So if we have 2 pyruvic acid, it will give us 2 NADH, right? And 2 carbon dioxide. And that's what we see in the model. During the Krebs cycle, the molecule of acetyl-CoA that was made will now enter. And the goal of the Krebs cycle is to remove as many electrons as possible so that way we can take them to the electron transport chain. So if we have a Krebs cycle, so let's start out with acetyl-CoA, 2 carbon. And then eventually this 2 carbon molecule from this 2 carbon molecule, we will bring it into the Krebs cycle. Right, so in this Krebs cycle, what is being released? ATP. NADH, and FADH2. And these molecules, our electron carriers, will guide the electrons to the membrane so that way the electrons are not bouncing around the cell. So they're guiding it to the electron transport chain. So let's look at our Krebs cycle, and I'll go over some of the points here again. At the beginning of the Krebs cycle, our 2 carbon acetyl-CoA will combine with oxaloacetic acid, which is 4 carbon. All right, so we have this, and the enzyme that will convert that will be citrate synthase. All right, so what was the role of the synthase? Synthase means to join. All right, so it's joining the 2 carbon with our 6 carbon molecule, and in order for the citrate synthase to work, the coenzyme A will help it. So with the help of CoA. So once we add the 2 carbon and the 4 carbon, we will get the 6 carbon citric acid, all right? How many carbon does citric acid have? 6, right? We have 6 dots. And citric acid gets converted into isocitric acid. If you look at the model, citric acid and isocitric acid, they look the same. But the name, the written name changes. So what enzyme is that? It is possibly an isomerase, right? Because we see visually, we can't see visually because we can't see the actual, we don't have the actual structural representation, but we can see that the name has changed. So same number of carbon, right? Same written number, but different molecule. Once isocitric acid is made, we have another enzyme that will convert it to alpha-ketoglutarate acid. Alpha-ketoglutarate acid will be converted by what enzyme? So we have kinase, dehydrogenase, lyases, synthase, right? Kinases. So in this case, it is a dehydrogenase. So your dehydrogenases will convert it, and it's specific dehydrogenase. But one thing we want to look for with our dehydrogenases, we want to see the NAD. So one thing that the dehydrogenase does is that it helps NAD to accept electrons from isocitric acid. So when NAD accepts the electrons, it is oxidized or reduced. So if it's accepting or gaining electrons, this means that NAD is being reduced. And when NAD is being reduced, isocitric acid is being oxidized, right? They have to happen at the same time, right? So our redox reactions, if something is oxidized, then something has to be reduced, right? And the H, remember the H represents our electron, right? So it has gained the electron. The other thing that happens in this step is the loss of carbon dioxide, right? So if you go from isocitric acid, eventually one of the carbon, we'll put a line through this one for representation, right, is gone. So alpha-ketoglutarate acid now becomes 5 carbon, right? Because carbon dioxide was lost as a waste product. And where does carbon dioxide go? All the carbon dioxide of the pathway will be removed from the cell or released. Right, so it will be released from cell. Carbon dioxide will be the waste product. In our 4th step, the alpha-ketoglutarate acid gets converted to succinyl-CoA. And similar thing happens, again, we see NAD, so this is a sign that we have another dehydrogenase, and the alpha-ketoglutarate acid is going to be oxidized, where NADH will be reduced. Okay, so again, our redox reactions. So NAD, when it picks up an electron, gains an electron, now becomes NADH. Right? And the carbon molecule is always going to be reduced. The carbon molecule that came from acetyl-CoA, right? So the same dots, right? The same dots here. And what happens in this step is that the alpha-ketoglutarate acid also loses another carbon and acquires CoA. So we see that CoA is coming in and carbon dioxide is leaving. So this molecule now is a 4 carbon with acetyl-CoA, which will help, with CoA, coenzyme A, the helper, which will help in the next step. Succinyl-CoA synthase will bind to succinyl-CoA in step 5 and convert it to succinic acid. In this step, what's being joined is the ADP with phosphate, so that way you can make ATP. Right? So synthase to join. right? And it's joining your ADP with your phosphate for phosphorylation. So the cell gains energy. Right, in step 6, succinic acid is converted to fumaric acid. Which enzyme is involved? So as I mentioned in the beginning of this lecture, your dehydrogenases, you'll see those involved with FAD and NADH, right? So what happens here? FAD now gains H, right? Electrons. So when it gains those, this means that this is being reduced, and then what is being oxidized? Your succinic acid, the carbon molecule which is coming from acetyl-CoA. In step 7, fumaric acid is converted to malic acid. Since the number of carbon looks the same before and after, the enzyme that does that is going to be isomerase. All right, so isomerase, again, the name changes, so it's indicating that the visual structure is changing, but still 4 dots before and after, 4 carbon. Right, so 4 carbon here, and still 4 carbon here. After step 7, malic acid is converted to oxaloacetic acid. Okay? So what is involved here? It has to be a dehydrogenase because we see NAD. Right? And NAD, since it's gaining electrons, right, that means again, similarly, NAD is going to be reduced, and malic acid will be oxidized. Remember, that has the carbon, the dots, from the beginning, right, from where we started from, with acetyl-CoA. And before acetyl-CoA was made, acetyl-CoA was glucose. So it all ties into oxidizing glucose, right, so we can get energy. So in this model here, we start out with 1 acetyl-CoA, but keep in mind that there were 2 acetyl-CoA that we started in this pathway. So if we have it, right, if we have it here, so I'll write it here, we started with 2 acetyl-CoA, and where did the acetyl-CoA come from? The 2-pyruvic acid. And before it was pyruvic acid, it was 1 glucose. So eventually, the 2 acetyl-CoA will give us what? Okay, so what does it give us? Well, since this is 1 acetyl-CoA, how much ATP would we have at the end of this step? Right, so if 1 acetyl-CoA gives us 1 ATP, that means after the Krebs cycle of 1 glucose, we will have 2 ATP. If 1 acetyl-CoA gives us 1, 2, 3 NADH, at the end of this pathway, how many NADH will we have? How many molecules? So we would have 6. Okay. And at the end of this pathway, we also make FADH2. Right? So 1 acetyl-CoA gives us 1 FADH2. 2 acetyl-CoAs will give us 2 FADH2. So at the end of this cycle, the Krebs cycle, we start to get these electron carriers. And the electron carriers of NADH and FADH2 will eventually make us lots of energy. So the goal is to extract these electrons so we can take them to the electron transport chain and get lots and lots of energy. And the cells will be happy. Right? They will get more energy than they started out with, finally. So after the Krebs cycle, and I wrote this down in total, you'll see that there are 6 NADH, 2 FADH2, and the carbon dioxide gets released, and 2 ATP are made. So to go over some of the steps, we have glycolysis, the intermediate step. We also have Krebs cycle. All right, so let's have a little summary. Right. So we see that there's ATP that's directly made. We also have NADH, which will carry electrons to the electron transport chain, along with FADH2. Right? And this is for aerobic respiration. Okay, so in total, for glycolysis, we said that in glycolysis, there was a net of 2 ATP at the end. Right? And that there was a total of 2 NADH that was made. How many FADH2? Well, it wasn't made at all. In the intermediate step, no ATP was made. There was NAD made. There was 2 that were made, right? 1 for each pyruvic acid. And no FADH. Okay? In Krebs cycle, however, everything was made, right? So at the end of the Krebs cycle, and we see it here, 2 ATP were made, 6 NADH, and then we have our 2 FADH2. So in total, if we were to total this up, at the end we have 4 ATP that are directly made in this pathway. Then we have our, if we add this up, 10 NADH, right? And for FADH2, we have 2. Right, so right away this all gets this energy, but this is the way because the NADH and FADH2 has to take the electrons, guide them to the electron transport chain. Okay, so this is our summary, and you see it here again, right? I told you 10 NADH, 2 FADH2, and they're taken to your electron transport chain, which is here, right? So just to summarize, to look at this again, in total, the cell will make 38 ATP, and that's the maximum ATP for prokaryotes. But what about eukaryotes, right? Eukaryotes make slightly less. Eukaryotes make how many? They only make 36 ATP. Right, so they make slightly less, and that's the maximum. It doesn't mean that all the time a cell will make that amount of energy, but that's the maximum that they're capable of making. And eukaryotes always make slightly less than prokaryotes. The electrons will be guided to the electron transport chain by NAD and FAD. Okay? So for the electron transport chain, you do need to know the major electron carriers in the membrane. So what are they? So we have cytochrome BC1, NADH dehydrogenase, and cytochrome oxidase. Okay, so our first one that we have, so I'll put a star next to it, NADH dehydrogenase. So maybe I'll put a star here. That one would be helpful so that we can see where it is. And then we have cytochrome BC1, which is in the middle, right? So cytochrome BC1, I'll put the star next to that next major complex, and then cytochrome oxidase complex, which is here, right? So those are the 3 major complexes that we have in the electron transport chain, all right? A chain of events. So one thing happens, and then the next thing is transferred and transferred, so that way something can happen. So at the beginning of the electron transport chain, NADH dehydrogenase collects the electrons from NADH. And it will remove those electrons, and it will move it across the membrane through these proteins, all right? So the 3 proteins, the 3 major proteins. When NADH comes to the membrane, NADH becomes NAD. So does this electron transfer chain, is it going to oxidize or reduce NADH? Well, if NADH is losing, right, it's losing the H, right, the electron, if it's losing the electron, then it's being oxidized at this point. Right? So NADH is being oxidized. And when it's oxidized, electrons are removed and carried across the membrane. When these electrons go across the membrane, it's going to activate each one of these proteins in the membrane. And we call them protein pumps. So once they're activated, then they start pushing protons outside. So at first, when the electrons move, 2 of these protons will go outside. And then as the electrons keep on moving, another 2 will go outside for NADH. And then another 2 will go outside for NADH. So altogether, 6 of these protons, a maximum of 6, can go outside of the membrane to change the gradient, change the amount of protons that are inside the cell compared to outside of the cell. Okay, so remember high protons here. All right, so I'll put arrows that it increases. And the electrons we said are going across. All right, so the electrons go across this way. And eventually all these electrons, remember, they're just moving across the membrane. So eventually what happens is that oxygen will collect them. So oxygen collects the electrons. Right, so the electrons, they have a place to go. Remember, the electrons were carried in by these electron carriers, and then they're brought to the chain. But if electrons just move about the cell, then eventually they will damage the cell components, including the membrane, DNA, and other structural components in the cell. So once oxygen collects the electrons, it will reduce oxygen into water. So when this goes to this, oxygen is reduced. Right? And water is inside the cell, outside the cell, so it's not going to have harm to the cell. So we said that NADH is collected here, right? And eventually NADH will lose the electrons will shuttle across and activate all these proteins, which we call protein pumps, and eventually we'll have up to 6 protons outside, high concentration of protons outside compared to what is inside the membrane. Right? So if we look at our model, I'll just label this is outside, this is inside, and this is our membrane. All right, so that is our membrane. So if we read here, the electron transport chain, this is what we're talking about, it receives the electrons. Remember the representation for the electrons, you may see hydrogen, and it will transport them to a terminal acceptor, which is reduced for aerobic respiration. So for NADH, as I mentioned, NADH starts here. But the NADH dehydrogenase only collects from NADH. It does not collect from FADH2. So where does FADH2 come in? It will come in here. And the succinate dehydrogenase will take its electrons. So when succinate comes in, when FADH2 comes in, it comes in at a later state of this electron transport chain. So since it comes in at a later state, it's only going to activate a certain part of these proton pumps. So when the electrons are removed, right, so this will become FAD plus, and when these electrons are removed, eventually they will too shuttle across the chain, collect by oxygen and make water. But when the proton pumps are activated, since FADH2 is collected later, only 2 protons will get pushed outside, and then another 2 later on in the chain, further down the chain. So in total, from NADH, we said we could get a maximum of 6 protons. But for FADH2, since it enters later on, we only get 4 protons. So less protons, less concentration outside of the membrane when we're talking about FADH2. All right, so this model you guys don't have, but you can look at it here. This is for NADH2 just to reinforce this. If we're starting out at the chain, so this will be the start of the chain, right? And then when we end, we're over here, right? The end of the chain, right? When oxygen is converted into water. So when we start out, we have lots of energy, and that energy is released so that way the electron pumps, right? NADH, dehydrogenase, cytochrome BC1, and cytochrome oxidase. These pumps can then pump out protons outside of the membrane. So let's look at them here. If we start out, NADH, right, we said is oxidized. The electrons will be released, and eventually they will flow across the electron transport chain. And as they flow, protons will be pumped out. We said for NADH2, 2 will be pumped out, then another 2 will be pumped out, and then another 2, so altogether 6 protons maximum for NADH when it comes to this site. The protons, right, since there's lots of them outside, high concentration outside, eventually these protons will be used to make ATP on the inside, all right? So make energy, and we'll talk about what is involved, what protein helps to convert or change this energy of protons into ATP, all right? So remember, NADH starts there's lots of energy that is using to work these proton pumps, and we get lots of energy, all right? But for FADH2, we don't start here, instead we're going to start over here. So we start a little bit later, not as much energy, so there's a loss of energy here. So when FADH2 comes to the electron transport chain, this is not working. All right, that proton pump is out of the picture. So FADH2 instead is, the electrons from FADH2 is collected by succinate dehydrogenase. And what will happen, these electrons eventually will activate our proton pumps. So we will get, as the electrons flow, we will get 2 protons outside, and then we'll get another 2, right? So only 4 protons, less than with NADH, and eventually those protons will make energy. So the rule is for every 2 protons, 1 ATP inside, right? So this will be inside the cell, right? So inside the cell. Okay, so what is allowing this transition from the high concentration of protons to ATP inside? All right, because electron transport chain is changing, right? It's converting these electrons into protons. So before we talk about it, just to talk about respiration a little bit further, we mostly focus on aerobic respiration, where oxygen is the final acceptor. It collects all the electrons so that way it doesn't bounce around the cell and damage it. But there's also anaerobic respiration. In anaerobic respiration, the terminal acceptor is not oxygen, but it is another inorganic molecule, right? And the inorganic molecules vary depending on the organism and what's their natural habitat. Later on, we'll see 2 organisms. One of those organisms, desulfovibrio, which can use sulfate as the terminal acceptor, right? So desulfovibrio, what shape is that? Right, we talked about shape. So vibrio is spiral. That would be a curved rod. So this is a bacteria that we'll talk about later on in our exam, in exam 2. And carbonate, this terminal acceptor is often used by many of our methanogens, right? And the methanogens, these are archaea. All right, so archaea. All right, so your archaea, and we'll see those examples, exam 2. Remember those names, desulfovibrio, metanogen. You won't be tested on them for this exam, but just to give you what's to come. All right, and they can perform anaerobic respiration. Also, anaerobic respiration yields less energy than aerobic respiration, right? So that's also the key difference. We're looking at differences. What makes them different? So just because there's no oxygen, just because oxygen is not present, doesn't mean that the cell cannot perform respiration. It may do it in another way using different terminal acceptors. One thing I do want to point out with this slide is that anaerobic respiration, so anaerobic respiration is not fermentation, right? So it's two different processes, right? Two different processes, so two different outcomes, right? So this has nothing to do with fermentation. Chemiosmosis is when the cell makes energy, right? So how is the cell going to make this energy? As I mentioned, the concentration of protons on the outside eventually will be used to make ATP on the inside. So what happens here is that our protons, right, will be pushed outside, and that concentration will be used to activate our ATP synthase, which will use those protons as energy to work this pump and start pumping ATP on the inside. So the rule is that one NADH will give us 6 protons on the outside. That is the maximum. It could be less. But if 6 protons are on the outside, then inside we get up to 3 ATP here. All right, but FADH2, remember, it starts a little bit later. It starts somewhere around here. And when FADH2 comes to the chain, 4 protons are pumped on the outside. So instead we get less ATP on the inside, only 2 ATP. All right, so the rule is, again, for every 2 protons, right? For every 2 protons, the equivalency will be 1 ATP. So that will be it. There are 2 types of phosphorylation that we often see in our pathway of respiration. So let's take a look at it so you know what it is. For substrate phosphorylation, this is what happens at step 7 and 10 of glycolysis. So step 10, right? So step 10 of glycolysis and step number 7, where our substrate, so this will be the substrate, will give a phosphate to ADP, and then eventually will make ATP, all right? So ADP, 2 phosphates. When it gains the phosphate from the substrate, now it is triphosphate, 3 phosphates. So again, ADP, or to correct it, or to make it clear, ADP has 2 phosphates, and ATP now has 3 because it acquired a phosphate from the substrate. So what enzyme does this transfer? We move the phosphate from here to here. What enzyme transfers phosphates? Right, so this will be a good clue. Like, let's say if you had this for the exam, a picture like this, you should know that this will be done by a kinase, right? Something like that. Right, so that should work. Substrate phosphorylation only produces a small amount of energy in the cell, right? When we're talking about respiration. So how is the other ATP made? It's made in oxidative phosphorylation, and oxidative phosphorylation is when the energy is generated through the electron transport chain. So remember, the electrons are coming from NADH or FADH2. Those electrons will go across the electron transport chain, right? And we have proton pumps that will push the protons outside, and this will create a high concentration of protons compared to a low concentration on the inside of the membrane. this change in gradient of high to low creates a lot of pressure, so that way the protons will come across this ATP synthase, this protein in the membrane, and it will activate it. So that way the ADP will collect the phosphate, and then it will become ATP. Okay, so that's how the ATP is made. It is made using the electron transport chain in addition to ATP synthase. Right, so in total, this is our summary of all of the energy that gets made in our transport chain. Remember, we have 2 ATP that were made in glycolysis, another 2 ATP that were made in the Krebs cycle. So altogether, that's 4 ATP. And then we had 10 NADH, right? And we said for every 1 NADH, for each NADH, how many ATP do we get? We get a total of 3, right? So 3 ATP, that will give us 30 ATP. And then for FADH2, there's 2 of them that is made throughout the pathway. So for every FADH2, we make less ATP, we only make 2 ATP. So altogether, 4 ATP will be made, all right? So if we add this up, from NADH we get 30 ATP, another 4 ATP from FADH2 when it goes to the electron transport chain, right? So this is what we're looking at, this number, and then the 4 ATP, all right? So if we add that together, 30, 4, and 4, we get the total of 38 ATP. Right, so where does this all occur? In different areas of the cell. Okay, so for the prokaryotes, very simple. Most of it will happen in so if you have your bacteria as a representation, right, most of it will happen in the plasma membrane. You would have your electron carriers, right, 1, 2, 3, the 3 major proteins, and then you'll have your ATP synthase here, right, so ATP will come in. So this is what it will be. So your simple bacterial cell, the electron transport chain, and ATP synthase is in the membrane. But with eukaryotic cells, of course, they're slightly larger. The glycolysis and the intermediate step happen in the cytoplasm, and then eventually what happens is that the process has to get transferred to the mitochondria. So with the mitochondria, where are these complexes? Well, same. The mitochondria look similar to the bacterial cell, so they are going to be pretty much along here as well. Right? So this would be our representation. So the Krebs cycle happens in the mitochondrial matrix, which we can say is kind of like the mitochondrial cytoplasm, the inside, right, of the mitochondria, and then the electron transport chain and the ATP synthase, that energy gets, so this will be where the energy comes in, ATP, right? So let's see if we can draw that a little bit better. So that's our cell. Let's make it about the same size of the bacteria, right? So that way our mitochondria has a better representation. And then this will be the ATP synthase, right? So the ATP will come in here. Okay? So the inner mitochondrial membrane will represent sort of like the plasma membrane of the mitochondria. Right? Remember, the mitochondria has a double membrane, an inner and an outer membrane. Right? So it's a little bit different as far as the setup. Okay, fermentation, as I mentioned, is different. Right? So it is a different process, so it's separate from what we have already learned. Right? So there's many benefits for fermentation. Fermentation can be used to make foods and alcohol. That's something that Pasteur did. Louis Pasteur showed us that our yeast can convert substances into alcohol. And when that happens, it tastes good, right? We like it, but then there's other bacteria that can convert alcohol into acetic acid. And what is acetic acid? It's vinegar, so it gives things a sour taste, and it doesn't taste so good. So we'll put that, all right? So it doesn't taste so great. So it can cause spoilage, it can cause damage to our material. So we do use fermentation a lot in our world today. So what is going on in fermentation? So this is your summary of fermentation. So the key differences, all right? So the key differences between respiration, remember you have aerobic and anaerobic respiration, which are sort of very similar, but fermentation is different, right? So fermentation occurs without the Krebs cycle and electron transport chain, right? If they're present in the cell, if those enzymes and proteins are present for the Krebs cycle and electron transport chain, the cell won't use them when they're performing fermentation. And usually fermentation will occur when there is no oxygen, all right? So the cells are not, maybe they're in a poor circumstance and they want to save their energy. So this will allow them to save energy by performing fermentation. It also uses organic molecules as the terminal acceptor, all right? Not inorganic, organic. So what are these terminal acceptors? We'll see it in the pathway here, all right? So this is our model for fermentation. So let's take a look at it. So for fermentation, what is usually broken down is a sugar. So glucose we'll use as a model. As far as lipids and proteins, they're broken down by respiration, and we'll see that in a little while. All right, so for fermentation, we start out with glucose, the sugar, and in 10 steps it's broken down into pyruvic acid, just like we saw in respiration. But what happens to pyruvic acid depends on each organism. Each organism has different enzymes, so they will do something different with pyruvic acid. Pyruvic acid, if we look at it, 1, 2, 3 carbon, can be converted to lactic acid. And lactic acid can be used to make things like yogurt and cheese and pickles, right? Many foods that we like. All right, so pyruvic acid, when it's converted to lactic acid, in order to do this, we have lactic acid dehydrogenase. And lactic acid dehydrogenase, again, we see the NAD, that's something that I told you to look out for the dehydrogenases. But what happens is that, in this case, if you look at it, 1, 2, 3, right? And you look here at the second carbon, you see C double bonded to O. But if we go down to lactic acid, what happens? We see more hydrogen or more electrons that have been added. So if we're adding electrons, if pyruvic acid is gaining electrons to become lactic acid, then pyruvic acid is going to be reduced. And when pyruvic acid is reduced, NADH is going to be oxidized. All right, so it's oxidized, NADH loses the H, right? Remember the H is the electron, and it becomes NAD plus, all right? So who's collecting the electrons in lactic acid fermentation? Lactic acid, right? They collected the electrons. They collected electrons around the second carbon, right? So this will be the terminal acceptor. For alcohol fermentation, it's slightly different, right? So some organisms, they may be able to make alcohol instead, and from pyruvic acid, they will have pyruvic carboxylase to remove a carbon. So we go from 3 carbon now to 2 carbon, right? Carboxylase remove carbon dioxide. Right? And we're talking about, let's say we're talking about making bread, right? We'll see that carbon dioxide can help to make the bread to rise, right? Fermentation. Okay, so once the 2 carbon molecules, maybe call this acetaldehyde, the acetaldehyde is then converted into ethanol by pyruvic dehydrogenase, all right? We see NAD again, NAD and NADH. So what happens, if you look at acetaldehyde, if you look at the first carbon, we only see one hydrogen. But when we make alcohol, we see two more is added, right? So there's three altogether here, only one hydrogen. So if acetaldehyde is gaining electrons, that means that acetaldehyde is being reduced. Right? And if we look at NAD loses H, right? The H is a representation for our electron. Then it becomes NAD, so the NADH is being oxidized. Different organisms, based on their genetics, have different fermentation patterns. So not all of the organisms do the same thing. However, during fermentation, the cells don't get 38 or 36 ATP. They only get 2 ATP for glucose, which is a very small amount, but it allows the cells to still make energy without having to use the entire pathway of Krebs cycle and the electron transport chain, which requires more energy, and it also requires the inorganic terminal electron acceptor to be present in that area. So let's look at the patterns of fermentation. Homolactic. Homo meaning that it only makes lactic acid. Right? So only makes lactic acid. Right? Streptococcus and streptobacillus. What shape is streptococcus? So streptococcus, coccus means round, and lactobacillus, bacillus means rod. Right? Both of these organisms are gram-positive. Right? So only lactic acid, homo, making one of the same thing. Right? Lactic acid can cause, I did mention food production, but it could also cause food spoilage, depending on the food, right? Every food is going to be different in the taste, right, that we're looking for. Heterolactic, hetero means mixed. All right, so in this case, these organisms are making lactic acid still, but they're making other things, mixed, all right? So two different or multiple, it doesn't have to be only two, it could be other things. So lactic acid plus other substances. Your E. coli and other enterics will be examples of in other enterics. So E. coli is an enteric, but we have other enterics in microbiology, and these are going to be found in the gut. Right? E. coli is in the gut and the colon. E. coli is gram-negative, and so are these other enterics that we're talking about. Saccharomyces was first discovered by Louis Pasteur, and this was a yeast, not a bacteria, a yeast, right? So we're talking about fungi that was used to make ethyl alcohol and carbon dioxide, and that's used for making beverages and also making bread. All right, so if we were to use, let's say, E. coli to make alcohol, it wouldn't taste good. Right, because that makes lactic acid and other stuff. So when you're fermenting a certain product, or a certain material, you're making food, you want to know what you're trying to make. You can't just use any fermenter. You have to use a specific fermenter. Propionic fermentation is done by Propionibacterium. This Propionibacterium we'll talk about it more on the second exam. But this is a gram-positive curved rod, and is going to make the holes that we see in Swiss cheese. In your lab, you will learn about fermentation testing and other biochemical testing. You will not be tested on it for this exam, so I will not go over this slide. But one thing I want to mention about biochemical test or fermentation is that you are looking to see if an organism contains a certain enzyme, right? So you're looking to see if an organism contains a certain enzyme. And based on whether or not the organism has an enzyme or not, you should see something different. So if one has a certain enzyme and the other organism does not, you will see a difference, all right? So that's what you'll be learning about biochemical. Biochemical meaning looking to see the presence or absence of enzymes in these cells. Lipid catabolism is the breakdown of our fats, right? Our lipids are fats. I did update this, right? Lipase, right? So lipase is going to break down lipids, and it will break down the lipids into its smaller components, which will be glycerol and fatty acid, right? So those are the two monomers that, or the two smaller units that make up lipids. Lipase catabolism is dependent on oxygen. It's usually dependent on oxygen, but I want to say mostly that it's dependent on respiration. Right, so lipids are not fermented. Right, so that's really what's important here. If we look at it for glycerol, glycerol is converted into DHAP, and we've seen this molecule DHAP before. In what process? Which part of the pathway? In glycolysis. So once it's converted into DHAP, it will enter along glycolysis, right, for about, let's say, 6 steps or so. And then eventually it will enter into the Krebs cycle, right, where it will be further broken down. And eventually, whatever electrons are made, those electrons will be brought to the electron transport chain. As far as fatty acids, they will be oxidized. We call that process beta oxidation, where this fatty acid, which has many carbons, remember the fatty acid, right, it has all these carbons, right? And all these carbons will carry electrons. So it will look something like that. You guys can go back to your notes on fatty acids, but we know that it has so many electrons. Right, we know that it has so many, so this will be your representation of a fatty acid, right? It has so many electrons, way more than glucose. But those electrons are eventually removed, the fatty acid is eventually broken down, its constituents enter into the Krebs cycle, and all these electrons of the fatty acid will be brought to the electron transport chain. So if we're talking about one fatty acid gives us about 336 ATP. But these triglycerides that we've talked about have 3 of them. All right, so that's almost, for 3 fatty acids, almost 1,000, right? Approximately 1,000 ATP molecules, right? So a lot more energy than glucose or one amino acid. For protein catabolism, proteins are broken down by proteases. All right. So these proteases, if you have your protein, so we start with our protein, they're extracellular or exoenzymes. Okay. And these exoenzymes, they go outside the cell. Okay. So some other exoenzymes I'll mention, you have sucrase, and sucrase degrades what? Sucrose. And degrades sucrose into glucose and fructose. And then you have the other one that I mentioned, lipase. So let's put other exoenzymes. Let's do something like that. And this will break down your lipids. So it will look like this. Right? So they will break them down outside the cell, and when they break them down, only the small parts come in. So if we're talking about sucrose, once sucrose is broken down outside by sucrase, which is an exoenzyme, then glucose and fructose come inside the cell. And for lipids are broken down outside. What comes into the cell? The smaller units of the fatty acid and your glycerol. For proteins, these are broken down outside the cell. Let's say if we have peptone or beef extract, which is high in protein, they will get broken down outside the cell, and the amino acids will enter the cell. Right? Eventually, the amino acid will get broken down. Deamination, D means removal of. So deamination is the removal of the amino group. Decarboxylation is the removal of which group? The carboxyl group. Then we have dehydrogenation, removal of the hydrogen. Desulfurization, removal of sulfur groups in some amino acid. If we look at this model here, right, it might look very confusing, but if you look along here, right, we saw oxaloacetate, citrate, isocitrate. So in the middle here, this circle is representing your Krebs cycle. Right, so that's what's going on in the Krebs cycle. And different amino acids enter in different parts of the pathway. So they might not generate the same amount of electrons. So if they don't generate the same amount of electrons, maybe you won't have, so you will not get the same amount of energy. So some of your amino acids, tryptophan, all right? So tryptophan is one of those amino acids. It's very high in a food that you eat often, or not often, well, around Thanksgiving, right? And that is turkey, right? Turkey is high in tryptophan. So tryptophan and some of these amino acids enter very early in the pathway, so it will generate lots of electrons and lots of energy. But if we look at something like aspartate, right, it enters later on in the pathway, so it will not give you as much energy for the cell, right? So different amino acids, again, enter in different places. Lastly, we will talk about anabolism. You know, anabolism is different from catabolism because we are now synthesizing molecules. So anabolism or biosynthesis, you're making, right? So in order to make something, well, first thing you need, let's say you need a nutrient, right? So your nutrient, let's say, is glucose, right? And glucose gets broken down. You get these building blocks. But in addition to building blocks, the cell also gets lots of ATP. So I'll draw my arrows meaning high or increasing ATP. And once we get this, the energy that is made in these building blocks then can fuel anabolism. All right, so this is what we have. Okay. So if we look at it, the building blocks that we've seen as our intermediates in the pathway, glucose 6-phosphate or glucose 6-phosphate is used to build things that have carbohydrates. So peptidoglycan, glycocalyx that has these carbohydrates, you will need the building blocks. But where do you get these building blocks from? From catabolism, right? And with energy, the cell can then build them up, synthesize these molecules that it needs. DHAP and acetyl-CoA, those building blocks are needed to make phospholipids. You can get them from breaking down your lipids, but you can also get DHAP and acetyl-CoA from breaking down sugars, right? If we talk about glucose catabolism, we saw that DHAP and acetyl-CoA were two building blocks that were made, right? And we can use that to make lipids that will become part of the cell membrane. And then the Krebs cycle intermediates, many of them are used to make proteins and enzymes. Right? And in order to take those Krebs cycle intermediates and convert them into amino acids for proteins and enzymes, we have the process of transamination. Right? So in transamination, the Krebs cycle intermediate or building block, so this is alpha ketoglutarate, is going to acquire an amino group. All right, so it will acquire an amino group. Okay, so it already has the carboxyl group. Now it will get this amino group. So this amino group eventually will attach here. And what you would get is your amino acid glutarate. And this amino acid can go on to making the material, the specific material you need for the cell.